The electron transport chain basically is going to use NADH and FADH2 to pass these high energy electrons from the actual molecule into an electron carrier protein inside our little electron transport chain. What it will do is generate an H plus gradient. And if you think back to diffusion and osmosis, diffusion says that things move from high concentration to low concentration. What we're going to do is we're going to generate on one side of a membrane a high concentration of these H plus ions so that they naturally want to move back across. And to get back across, what they've got to do is move through this guy named ATP synthase, which is a turbine-like enzyme that actually will take ADP and force it into close contact with ATP, or with another phosphate, rather, that creates an ATP. So these hydrogen ions moving through this protein because of a diffusion pressure will actually generate ATP from an ADP and a phosphate. This image, although it's got a lot going on, kind of highlights that for us. Let's start it out nice and slow by taking a look at what we're looking at. Here's a mitochondria, and what we're looking at is we're zooming in on this little spot right here. You notice I've got two X's drawn. The red X is over here, the black X is over here. That should kind of give you reference of what we're talking about. So I'm looking at this membrane, and I've taken the single line, and I've zoomed in so much so that here's the line, and here's the actual space that's there. So we're looking right at that line as I've zoomed in. You'll notice that I've drawn in blue, I've drawn these electron carrier proteins. I've not named them because it really isn't important for our class that you know the name, other than the fact that in blue are our electron carrier proteins. And what's going to happen is these electron carriers that we've been using before are going to drop their electrons off at the electron carrier proteins. And then these proteins will be used to generate a hydrogen ion gradient. I've used different colors to highlight each of our electron carriers. So let's take a look. First, we're going to look at NADH. It's an electron carrier that will be dropped off at the first electron carrier protein. And so from carrier protein to carrier protein to carrier protein, that electron is going to get passed. So you can actually see its progress from 1 through 2, out 3, and then out the end right here. That's pretty important because it's going to pass that electron down a cascade effect. This is going to allow your body to slowly release the energy. Each time the electron is passed through one of these carrier proteins, it is actually going to release a hydrogen from this side and pump it to the other side. So what you end up with is that for each of our NADHs, you're going to move a total of 3 hydrogen ions. And what we'll end up with is a really high concentration over here that naturally wants to flow back across. But the only way it can get through is ATP synthase. So NADH will move through all 3 carriers, it'll move 3 hydrogen ions, and each time it does so, it needs this oxygen right here to collect the electron. It's going to use hydrogen ions over in this area to finish the formation of water. That water is a waste product with respect to cellular respiration. So NADH pumps 3 hydrogen ions, and then each of those hydrogen ions are going to move through ATP synthase, yielding 1 ATP apiece. So that the things that I have marked in black are all NADH. It's dropped off at the first electron carrier protein. The electrons move through all 3. It's going to move 3 hydrogen ions. Those 3 hydrogen ions will then move back through the ATP synthase molecule, and each one of them generates their own ATP molecule. That's NADH. FADH2 is slightly less effective. However, you'll notice that FADH2 here enters into this intermediate electron carrier protein, then goes through the second and third, and then eventually exits. Again, names aren't necessarily important to us. Just simply know that it bypasses this first one. What that means is that your FADH2 only moves 2 hydrogen ions. Because it only moves 2 hydrogen ions, you're only going to get 2 ATP out of it, which means that NADH will give you more ATP using ATP synthase, and this particular process to give you less ATP. Either way, all of the NADHs and FADH2s we built up will generate about 32 ATP molecules with a glucose. What that means is that the vast majority of our energy is made here, and the vast majority of our energy is absolutely dependent on the presence of oxygen. Again, this electron transport chain is a little complicated, but if you follow the colors that are here, I think you'll be able to see what's going on. We're looking at a mitochondria. NADH and FADH2 are going to drop their electrons off to these 3 electron carrier proteins. They'll move through them to pump hydrogen ions, which make ATP. Again, like I've said, the energy totals per glucose are about 36 ATP. The vast majority of those coming right here in the electron transport chain, and then 4 others coming in glycolysis and Krebs cycle split respectively. This represents about 36% of the total energy of glucose, and the rest is actually released as heat. So when you're running around or doing exercise and your body gets hot, it's because it's going through this process, which is releasing a lot of energy as heat. Fermentation happens when there is no oxygen presence. And simply put, fermentation, its main goal is to get back NAD+. Because what your body wants is energy, but for it to go through glucose, for it to go through glycolysis and break down glucose, it actually has to have NAD+. You might ask yourself, well, why doesn't it need to regenerate ADPs? Remember that ATP is a molecule that can be readily used in the cell, so regenerating ADPs is not a problem at all. However, NAD+s really only get used in the electron transport chain. So we got to find a way to get that back. And what fermentation does for us is get those NAD+s back. Our bodies go through lactic acid fermentation. This is what causes muscle soreness after severe or heavy exercise. And basically what that means is that these pyruvic acids are then turned into 2 lactic acids. And what we'll do is we'll return that NAD+ to allow glycolysis to continue. In other organisms, such as yeast, they'll go through alcoholic fermentation. It has an extra byproduct of CO2, so it'll actually produce carbon dioxide. And so if you have breads that have little holes in them, which most do, those little holes are generated because of yeast rising the bread and stretching it, and the holes are carbon dioxide that's captured. If you ever made bread, you want to be careful not to deflate it by breaking and allowing the CO2 to exit, because then your bread won't expand right. The other thing that yeast will actually produce is ethyl alcohol, which actually is removed from the bread by baking it, so you're not going to get it there. But ethyl alcohol is also used, or this process is used, in the generation of some wines and spirits. Again, focus of fermentation is quite simple. You want to get these NADP pluses back so you can continue going through glycolysis.